to do. The plan is to talk to you about how you can kill grandma, or at least make her almost stop breathing. Do the use of peripheral chemoreceptors. Then a little urine analysis lab where you guys can analyze your own urine, centrifuge it, and look for some crystals that might be in there, or at least look for the, what's the type of cell that lines the bladder again? What epithelium was that? Transitional, so you can see any of those. You remember how you recognize transitional epithelium? The one big dead giveaway, you see it, you quit thinking about it?
The two nucleus.
The binucleate, you might find some binucleated cells in your sample, right? Or lots of simple squamous from the urethra itself. And then look for your normal glucose, pH, and ketone values in there. And we'll talk about how to do that when we're done with this little discussion. Then after that's done, we'll turn you guys loose and let you work on whatever it is you need to work on for your renal anatomy or respiratory stuff. So last Wednesday we talked about the idea of the central chemoreceptor, otherwise known as the hydrogen ion sensor, located in the medulla, in the cerebrospinal fluid environment of your medulla. And it was detecting levels of hydrogen ions in your cerebrospinal fluid due to the diffusion of CO2 into that fluid, doing your favorite chemical reaction of CO2 plus H2O, giving us hydrogen ions. Your central chemoreceptor is what's dictating all of you and all of your normal breathing. That's what is your primary driver right now. So when you hold your breath and CO2 levels increase, right, you're thinking hydrogen ion levels and the cerebrospinal fluid will increase. Because remember, it isn't the hydrogen in your blood that those sensors are detecting. Because that hydrogen can't leave the capillary and get into the cerebrospinal fluid. So that hydrogen had to come from the reaction of CO2 and water of your cerebrospinal fluid. Under normal conditions, your hydrogen ion levels increase. That means more hydrogen is going to the hydrogen ion sensor, and you breathe more. Problem solved. But under prolonged levels of elevated levels of CO2, whether it's because you're in high altitude for the first time in your life and living there from now on, or you have pulmonary disease, or you have COPD, emphysema, for whatever reason, prolonged exposure to elevated levels of CO2 will give you prolonged exposure to elevated levels of hydrogen ions, which means prolonged decreased pH in your cerebrospinal fluid. The ependymal cells don't like that. They release a buffer. The buffer then sequesters, binds to the hydrogen that normally talks to the hydrogen ion sensor. If I'm using that up to make carbonic acid, I don't have those hydrogens to go to the sensor. So how does the sensor know to make me breathe faster with those elevated levels of CO2? It doesn't. We have a fallback. We have this. Because this is your peripheral chemoreceptor. Remember, they're going to be in the carotids and the aorta. Remember, we talked about how they will respond to significant changes in O2. But if you say significant changes in O2, that's a vague answer. Because does John mean elevated levels of O2 or decreased levels? Remember, it was decreased. Do you remember the actual partial pressure value that if we get below this value, these guys are going to kick in? It was a PO2 of 60 or less, right? That's the significant decrease in O2 we're talking about. PO2 of 60 or less, these guys get really active. Now we got to figure out how these guys do what they do. So I need blue. What I drew up there for you was one of our cells of the peripheral chemoreceptor. Yeah, we'll hold that for a minute. And this cell is called a glomus. That's a G, that's 6. Hang on here. Be careful, it's not a glomerulus. It's a glomus. And that's the name of the cell that is a peripheral chemoreceptor. If I said name the cell, you call it a glomus. If I say what kind of a cell is it, it's a peripheral chemoreceptor. Now, in our blood, we've got O2. So there's O2, and it'll diffuse into the tissues. And I've got all of these chemical gated potassium channels. These are all chemical gated potassium channels. And what does that tell me? Well, it's a gated potassium channel that responds to what? A chemical. That's why they're called chemical gated potassium channels. These guys down here in orange are a voltage-gated calcium channel. And what do they respond to? Voltage. That's why they're called voltage-gated. Remember, when you hear the word gated, that means a stimulus is required to make them open up. Otherwise, they're going to be closed. So what would make a chemical gated potassium channel open? That's when O2 could bind to that channel. So when it has O2 attached to it, those channels are actually going to open up. I'll use a blue lid to represent the fact that they've opened due to oxygen. And if that's true, then the potassium can leave through those open channels. If potassium leaves a cell, what happens to that cell's membrane potential? It becomes more negative, right? So we're going to get a more negative, right, membrane potential. Now, the idea then is going to be, well, what if we don't have oxygen attaching to these chemical gated potassium channels? Well, we can see what happens when oxygen is attached to them, right? They open up. That's the chemical they're responding to. That's their stimulus, O2. But if I don't have O2, the absence of O2 means that these guys can't be opened. And if they can't be opened, can potassium leave through them? Nope. The potassium can't leave, right? So we're going to can't get out, can't get out. And potassium can't leave. Do we get a buildup of positive charge inside the cell maybe? So then we can have the cell membrane become more positive. Now, anytime membranes become more positive, we use the word depolarization. It doesn't necessarily mean action potential, but to depolarize something means to become more positive. So we can become, right, more positive. All these positive charges, right, more positive membrane potential. And it becomes positive enough. What's that going to do to a voltage? What does a voltage-gated channel need to open up? It needs a threshold of some kind, right? It needs some value of positivity showing up. So we're going to have enough positive charge to make these guys open up. If I can open up voltage-gated calcium channels, who's going to come in? Calcium. Calcium. And why is calcium coming into the cell? You guys should know this. This is your trivial pursuit. This has to be in your thoughts the day you die of oil. Oh God, there's Moffat again. All right? Things move due to concentration gradients, right? The reason calcium is coming in is due to a concentration gradient. Not because you opened the door. Right? Because there could be no calcium out here. Right? You just gave it the ability to follow its diffusion path. So the calcium that's out here now can follow, can now follow its concentration gradient into the cell, and the more voltage-gated calciums are open, the more calcium can come into the cell. And you're going, yeah, big freaking deal, who cares? Well, these are vesicles full of neurotransmitters, and I need to be able to release them. You need to remember that releasing neurotransmitters is a calcium-dependent process. Calcium must attach to the vesicles. And once the calcium attaches, that allows the vesicle, right, to kind of come down here, to move down. And I'm going to show one now actually doing exocytosis. It actually fuses with the membrane because they're both made of phospholipid bilayers. And now I get to do exocytosis of my neurotransmitters, landing on a neuron, and I get to increase action potential traveling down the length of my neuron. I breathe faster. Okay, so now let's look at this, right? When O2 was plenty, we had lots of O2. I started opening up these potassium channels. Potassium was leaving. We became more negative, right? We weren't having calcium come in. I wasn't triggering the release of neurotransmitters to make me breathe faster, because I already had enough oxygen apparently that I didn't need to breathe faster. But if I start diminishing or decreasing the levels of O2, maybe we'll kind of indicate that since we're talking about these guys. So with decreased PO2s, right, there's not oxygen attaching to these. So no O2 binding. Ooh, wow, I'm keeping that one. No O2 binding to chemical gated potassium channel. Right? Therefore, I'm running out of room, closes. And if it closes, potassium can't leave. Potassium can't leave, cell depolarizes. If the cell depolarizes, we open up voltage-gated calcium channels. Calcium comes in because we didn't have enough oxygen. So apparently I need to ventilate more. And so the more channels that are not open, the more potassium stays. The more voltage-gated calcium channels are open, the more calcium comes in, the more neurotransmitters are released, the more I ventilate because I'm low in O2. So now I come in, right? I go visit grandma, and I realize that Nana looks a little peckish and also a little cyanotic. So I have them bring in some lousy Jell-O and hospital food. But I want to help her out. I'm also starving, haven't had breakfast, lunch, or dinner yet. So I'm going to go to the cafeteria, but I realize that she probably could use maybe a little more oxygen. I don't know any better. I'm just trying to be nice and loving. So I raise her O2 levels. So I look at it going, God, a lousy frickin' 1.5. What the fuck are they? What are they doing? Sorry. Like, okay, that's not good. They always say more is better. So why not 3, 4.5? Why not 6 liters a minute? Right? That's a good flow. She'll be happy. Right? And I'm going to go to lunch. And I come back and maybe she's in respiratory failure. What the hell? All I did is give her oxygen. Well, all I did is trick her peripheral chemoreceptors into thinking she had lots of oxygen. So the more O2 that was in her blood, the more O2 diffuses into these guys. You gotta remember, when you're on O2, you're not on a ventilator. They're just pumping O2 up your nose. You gotta breathe deep. You gotta inhale it. Well, to breathe, I have to ventilate. I'm ventilating because these guys are telling me to ventilate. So even though there's O2 going up my nose, it's not necessarily going into my lungs. So you think about what they're giving you. They're not giving you air up your nose. What are they giving you? Oxygen. So what is in an oxygen tank? Oxygen. What's the percent of oxygen in an oxygen tank? 5%. All right. And so how do you calculate partial pressures? Atmosphere times percent. Well, normally the percent is 0.21, right? But now what's the percent? 1. 760 times 1 is a partial pressure of 760. Right? But we can't dissolve all that oxygen, but you're making sure that you have the maximum saturation level of your plasma and maximum saturation of the blood as it goes by the lungs. So the lungs are thinking, God, we're doing a fantastic job here. Maybe we only need to contract the diaphragm once every 5 hours because we have so much oxygen in our blood. That's why you give lots of O2, right? That's not good. And so the more O2 you give, the more these channels open up, the less calcium comes in, the less neurotransmitters are released, the less you ventilate.
So if they decided they wanted to increase the oxygen, the medical people who know what they're doing, they would presumably do it really gradually so that the cerebrospinal fluid bicarbonate levels would drop commensurately, or?
Well, so what I was taught,
just a certain amount they can give, and after that it's just the best you can do.
It's the best you can do. So what I was taught a long, long time ago by one of our physicians teaching me advanced physiology in my first undergrad was, a lot of times doctors are between a rock and a hard place, or medical sciences. You need more O2 because your PO2 is at 50. Should be at 100. All right. But then we realized that if I give you O2, I'm tricking these things into thinking you're ventilating quite well. So I'm between a rock and a hard place. I have to find the happy medium of give you more O2, but not so much to actually make these guys think that you don't need to breathe whatsoever. So there's that, you have to sort of, have to spend money to make money, Kind of an idea? I got to do a little harm to do a lot of benefit. But I can't do a lot of harm for a lot of benefit, because the more harm I do, actually the more dangerous it becomes. So that's why they have these standing orders. If you ever work in a pulmonary unit, right, and someone has emphysema or some other COPD issue, probably a standing order, no more than 1.5 liters a minute. Because if you give more, this begins to kick in. And the more you give, the more it kicks in. And the more it kicks in, the less you ventilate. And the less you ventilate, you're getting those gases to build up. Now your CO2 is going to go even higher than it is. And your ability to get O2 is going to get even less. That's how you can have the perfect Alfred Hitchcock murder. Because how do you detect levels of oxygen? You know, kind of cool. No, it's not.
Just to make sure, that would be the phrenic nerve. It's just
Well, that's a nerve from the chemoreceptors up to the DRG and the VRG. All right. Okay, so now when you look at this, now it'll make a little more sense. This came from an author known as Silverthorne. I really like how she talks about things. It's always about cause and effect. So there's your low PO2, right? The low PO2, and that's the hard part, is I always think, oh, she's showing me an oxygen molecule, and that makes the channel close. Like, no, what she means is the absence of an oxygen molecule makes the channel close, leading to depolarization, opening up voltage-gated calcium channels. Calcium comes in, lands on the vesicles, exocytosis, more action potential signal to the medulla centers, which is your DRG and your VRG, and I breathe more. It's just a nice little chart. Now, I don't remember exactly what I put. I think I just gave you 2 slides in that PowerPoint. You guys just had this one and this one?
Yeah.
Or did you guys have this as well?
No.
Okay. Yeah.
Is that one, the other one?
Well, it's the same thing that we did, but it's more of a generalized one, so don't look at it.
Keep it up.
No!
I want to take a picture.
No, I don't want to take a picture of it. It's too big. Okay? It's a concept map for how we actually do what we did on the whiteboard.
It's the same thing.
It's the same thing, but not nearly as detailed.
Okay.
It doesn't mention buffers by ependymal cells either.
Oh.
Okay, that's why I don't want you looking at it. Okay, so photograph this. I'm going to turn that off. Right? Urine analysis to find out what is in your urine to a certain extent. I'm not going to make you guys do this, right? But it's kind of fun to look at. You're going to be doing 2 things. One, taking a urine sample, so you get to pee in the cup. You're then going to be taking these little analysis sticks. And here's how you go about doing this if I were to do it. All right, I'm going to take out a urine analysis stick. And I'm going to put the lid back on. And I'm going to have my sample of urine in a cup. So this will be my cup of urine. And I'm going to take this, I'm going to stick it all the way in, so I submerge all of my little tabs, all my little test pads into the actual liquid of urine. You got to just work your way in there. Now it's all in the urine. Now, do not do this. And you got to think about, why is John saying this? There must be a reason why John is saying this. You take it out, right, and I've got this urine, right? Do not flick it. Right? There's a reason why I have to say this, okay? You just kind of tap it off to the side if you need to, right? Now, all of these are covered in urine, okay? Now, when you look at the sides of this, and I'll find a website that has this, so it's a little bigger to see perhaps, but you realize we have all these little tabs, and it says like 120S, 60S, 60S, 60, 60, 60, 40S, 30S. S stands for seconds. That's how long you wait to look at that part of the tab. So we go from 30 seconds up to 2 minutes in time. Right? After you take it out. So the very first blue one right here, the very first one is a know-nothing tab. Right? That very first blue one is my glucose. And then they all correspond to the various colors up along in here. And you just kind of go along and figure out where the color is. SG, that you have on here, right, is the third one up. That is specific gravity. That's, in other words, kind of a measurement of the dissolved particles inside of your liquid. Your pH one is quite variable. If you had a really high protein lunch, you're going to have a rather acidic urine. If you had more of a plant-based lunch, probably have maybe more of an alkaline urine. And so meals will quite quickly, readily cause significant changes in your pH with this. When you're done measuring your little tabs over here and realizing what your values are, this goes in the red can, right? Biohazard one. Just drop that little puppy in there. Then you're going to, if you want to, which that's kind of cool to do, you're going to take off our little pipette dish here. You're going to carefully, maybe over the sink, just in case you're not a good pourer, right? You're going to pour enough urine in here, either A, to get rid of it all, or B, get up to about 2/3 full. All right? Then you're going to put your initials on here. And when we get a bunch of these, we're going to put them in the centrifuge, right? And we're going to spin them for about 10 minutes, right? Like doing a hematocrit, right? And we're going to be pulling all the solids to the bottom, right? Your precipitates in chemistry. We're going to aliquate off all the liquid, the supernatant, and just take your pipette, go all the way down to the bottom, and grab some solid stuff. And suck up that solid stuff. So before you actually suck the solid stuff up, get the microscopes out. Have them already set up. All right? I'm going to suck up my solids, and then you're going to take your microscope slides, and you put a drop of your supernatant, right, onto, or precipitate I should say, onto your microscope slide. Add a drop of this material onto, then add a cover slip to it. All right? Then look for the cells and structures you can find inside of this little material. Be careful of the staining substances. When you're done, the slides and cover slips will go in the glass container. Right? When you're done with your urine sample, it can go down the sink because it all ends up in the same place, right? Water treatment plant, right? Paper cups you can crush and put in here as well with those. We have some books for you guys to look at in terms of what you're actually looking at. Apparently they don't change the information so much because this was printed in like 1980. You know, so I guess blood cells haven't changed a whole lot, right? Simple squamous epithelium hasn't changed a whole lot. Not a whole lot of need to do it. But what you're looking for the most part, is when you get here, most of this is not relevant to us at all, but there are some photographs of some crystals that you might find in your urine, and you probably won't find them at all, but in case you use some calcium carbonate crystals and such. But the idea of what would be normal, for example, in terms of the blood cells, not blood cells, pardon me, skin cells, epithelial cells, are these guys here. You'll find the simple squamous epithelium lining. We also find transitional epithelial cells. You'll find probably lots of these in here, and probably very few, if any at all, of the various crystals that are found in the back of this. Then you can work on your renal anatomy, right? And I do suggest that at some point in the next day or so that you watch the video I sent, right? And don't talk to your friends, but see if you can find the things that are wrong. There's some like blatant things that are wrong. Now I really like the guy who does these, right? He's really smart, he's really good, but everybody makes boo-boos, and this is why you don't go to the internet. Because if you were to learn from him, 99% is great, but the few things that are like wrong are like, dude, were you thinking what you just said? Hello? Then you're going to learn something incorrectly. See if you can spot the things that are wrong. It's a nice way of doing a little self-assessment, right? If you watch the others of his videos, A, keep it to yourself. Don't talk to others about the things that are in there that are really cool, that are not relevant to class. Because he gets into a nice 300, 400 level of biochemistry. Right? He gets in a nice, good depth. A lot of what he talks about, we have covered. Right? But it's also more than we have covered. So don't worry about what he does, what we have not done. So maybe when the unit is over, then go watch his videos for some extra material. But he does a really good job. All right, turn you guys loose. Happy peeing, happy micturating. Let's measure your pH values and your cell counts.